Hello, this is Dr. Dan Guerra reporting to you from VeraMed Studios in the inland Pacific Northwest. Today, we're going to cover a voluminous topic that I've been working on for some time, hence the towering effect of this dead standing Douglas fir tree that you're looking at in the middle of the picture. It's a tree that I eventually dropped with my chainsaw, affectionately known as Tangerine. But I digress. Let me get started with this talk, and we will get it recorded. Today is the 30th November, 2018, so we're almost entering the Christmas holiday season. So let's get going here. All right. I'm titling this The Diabetes of Diabetes as a Disease of Lipid Metabolism, or how does GTPase mediating GLUT4 endosomal trafficking mechanism associate with obesity-linked dysfunctional lipid homeostasis? You can choose which title you prefer. That's me, Dr. Dan Guerra, and that's that tree behind me there after I cut it with that chainsaw you see right behind my head. Anyway, let's get started. This is VeraMed, company that I co-own, and what we do is we verify the evidence and medical scientific research that comes from the refereed literature, that is published literature that you find in journals. That's my Facebook page where you can find all of these talks that I have so far recorded, and there are probably somewhere around 40 of them. And there is our website, www.veramed.com. This is my personal email address. You can contact me there to discuss VeraMed and discuss the topic and concentration of any of the talks I give. All right, let's get started with a introduction, which is usually a good place to start. AKT, which is a kinase, and AMP kinase, which obviously is a kinase, AMPK, regulate a protein called TBC1D1 through phosphorylation and its interaction with cytosolic tail of an insulin-regulated aminopeptidase, and that's called IRAP. So that's the acronym for that protein. So I know it's a mouthful, and I know there's a lot of things that probably, until after you've listened to my entire talk, you will know very little about unless you are indeed in this field. This paper was first put out on Journal of Biological Chemistry in October, so it's a couple months old now, and that was when it was in press. Now it is actually in, I think, the November 23rd published edition of JBC, 2018, of course. So let's get into the background. TBC1D1 is an insulin-mediating member of the RAB GTPase activating proteins, or called RAB gaps. They play an essential role in GLUT4 translocation. Now, GLUT4 is the glucose transporter, isoform 4, which is common in skeletal muscle in man. It's also found in, for example, cardiac muscle. So, but right now we're going to be emphasizing, at least for the most of the talk, skeletal muscle. It's how insulin-dependent glucose uptake occurs in the muscle. Because of the association of TBC1D1, it likely is linked to obesity and the occurrence of type 2 diabetes. Okay, that's because it regulates glucose uptake in skeletal muscle. Skeletal muscle is a huge homeostatic reservoir and interlocuting regulator of glucose homeostasis in the body, as well as lipid homeostasis. Something is often overlooked because normally you think about skeletal muscle, you think about contraction, relaxation, physical work, and things like that. Now, TBC1D1 has two amino terminal phosphotyrosine binding domains, they're called PTBs. It also has a calmodulin binding domain, CBD, and a C-terminal catalytic domain, which is called the RAB gap. Again, that's the activating protein. The RAB gap domain promotes GTP hydrolysis in its substrate, thus causing the conversion of the inactive GDP-bound isoform of the GTPases. Okay, so that's actually the substrate for this protein. So there's an arginine finger. It means it's rich in arginine. The sequence is rich in the amino acid arginine, located in that gap domain. In fact, arginine 854 in the mouse protein, for example. And it is required, that arginine, for full catalytic activity of the Rab gaps. Now, if it's the arginine, if you know the R group for arginine, it doesn't get phosphorylated. So that's what we're talking about here. Remember, this is a phosphorylation paper. The human genome contains about 70 Rab GTPases. Now, again, the GTPases are the target of the Rab gap. And in this case, the TBC1D1 we're talking about. There's 70 potential substrates. And there's more than 50 putative Rab GTPase activating proteins. So you can imagine the complex mathematical interaction there. You have 50 different activating proteins, right, which cause that GTP hydrolysis on some 70 substrates. So there's a lot to sort out here. It's not sorted out at all quite yet. All right. Human genome contains 70, as I said, GTPase and 50 Rab GTPase activating proteins. That was a paper published now a while back in Nature Cell Biology back in 2005. So it's been well established. Rab gap domains display specificity towards the following of the Rab proteins, the Rab GTPases. Okay, Rab8A, Rab10, Rab14. Okay, and those three proteins specifically are involved in GLUT4 trafficking from the endosomal compartment to the plasma membrane so that you get insulin-dependent glucose uptake in skeletal muscle. Now, TBC1D1 is a downstream target, that protein itself now, the activating protein, for two canonical kinases in the cell, which we've talked about a whole lot throughout several of my lectures, my video lectures. You can go back and determine where these proteins show up. They show up everywhere. They are absolutely common kinases in all kinds of biological activity. Too many to even account for here. Anyway, that protein is yet another target for those two kinases, the AKT and the AMP kinase. Now, I'll mention the AMP kinase in a little bit more detail later on, but the AMP kinase basically is triggered when adenosine monophosphate levels increase, which means the energy charge of the cell is being diminished. And if you're talking about skeletal muscle, that means after lots of contraction and relaxation cycles, where a lot of the ATP is burned up. That's when that kinase kicks in. Makes sense that it would control a protein that controls GLUT4 uptake, because that ultimately will then allow for insulin-dependent glucose uptake into the muscle. If it's regulating at that level, right? Of course. Phosphorylation of TBC1D1 on a threonine, that does definitely get phosphorylated because of its R group, OH group, by AKT, that kinase, in response to insulin, and serine 231, another amino acid that's phosphorylated, free hydroxyl group, by the AMP kinase, in response to muscle contraction. So that one's tuned up by muscle contraction, as I just mentioned. And subsequently, another protein called, I know this is terrible, but that's what it's called, 14-3-3, or the 14-3-3, we affectionately call it. And all that is essential for GLUT4 translocation. Okay, all of that. All right. Here's a good model of that. Without insulin, okay, this is what you see. There's the plasma membrane. There is a vesicle, a storage vesicle for GLUT4, GSV, GLUT4 storage vesicle. All right. Now, what happens here when insulin is not synthesized in response to high blood glucose, okay, in a homeostatic mode when glucose levels are depressed, you have this kind of situation. You have a PTB protein associated with a protein called AS160. And in this instance, the GAP protein, okay, that TBC protein we're talking about, is on, okay? And because it's on, what you have is a Rab GDP, right? You have a Rab GDP. So you don't have GTP in there because this enzyme is turned on, and the activating protein is a GTPase activity. So whatever cargo proteins are associated with it that are embedded in the endosome, this vesicle, along with GLUT4, are going to reside in that endosomal complex. Now, what you get with this system when the insulin is not signaling is you get what's called vesicle sampling, where the vesicle rises to the surface in this native state, some GLUT4 is translocated and some glucose is taken up in a non-insulin dependent pathway, as you can tell. Notice that the AKT enzyme is inactive here. There's another protein called the RAB effector, which we'll talk about in a moment. Now, when insulin is secreted by the pancreas, beta cells of the pancreas, this is what happens. This is very important. You get a alteration of the membrane, the plasma membrane. And we won't go into the detail exactly what's going on with the lipids there, but there's something very spectacular. The AKT is recruited to the membrane. Okay, recruited to the membrane. Now it's active. Okay, and it's going to, because it's phosphorylated, it's going to block a lot of this older activity that was occurring here. Okay, and when that happens, the PTB will still be bound, but you're going to get a series of phosphorylations because of the AKT. Okay, and those phosphorylations are going to enhance the binding of the protein 1433, excuse me. And in that process, the GAP protein, that's the GTPase activating protein, is turned off. Okay, now, but because it's turned off, RAB GTP builds up, and that's the active form of that protein. It can then interact with the RAB effector, and it can cause this entire vesicle to fuse to the plasma membrane. Rotate that vesicle right into that membrane, getting lipid-lipid fusion. And that's how you get GLUT4 to the surface, and that's insulin-dependent glucose uptake, ultimately, in skeletal muscle. This came from a paper published in Molecular Cellular Biology, 2012, as you can see. Now, here's yet another cartoon. I'm going to show you a couple cartoons at the beginning to get you aware of this. Now, there's more complexity than that. There's also a germination that has to occur, okay? So let's just take a look at this. Here's your active RAB GTPase, which is essential for GLUT4 translocation, insulin dependent. Okay. Now, what can happen to it? The RAB gap can hydrolyze the GTP, giving you the RAB GDP, which is inactive. Okay. Now, likewise, the RAB GDP can be exchanged out by the GEF. That's the exchange factor for the guanylate residue that is bound to the RAB. So it completely removes GTP and loads up GDP, making it inactive, but giving you the donor membrane in its native state. Likewise, the RAB gap can turn back on this pathway and synthesize the RAB GTP. Okay, so this is the process that you get. This is in a metastable state. Now, when you are in the inactive GDP form, that protein can be picked up by the rep. Let's go back over here. Cycle involves exchange whereby a conformation between the active GDP bound and inactive GDP bound form occurs. The balance between the two isoforms is finally regulated by the RAB GTPase. As you can see, it's coming in here, it's feeding into there, and it's also feeding into there. All right? Okay. And the RAB guanine nucleotide exchange factors, likewise working in both directions. Okay. When activated by GTP binding, like there, they recruit step-specific effector proteins. Okay, that's what's going to happen when they're functioning, like we saw on the last cartoon slide, which may have a role in linking vesicle and target membranes to the cytoskeleton. These are going to be proteins that are going to help dock and initiate the fusion of the vesicle with the plasma membrane via interaction with the cytoskeleton. Effectors dissociate when RABs convert to the GDP-bound state, okay, as you can understand from the nomenclature, and the inactive RAB can then be removed from the membrane, shown here, via the RAB GDP dissociation inhibitor. So this protein is part of the cycle too. Now over here, I want you to take a look. In order for this protein to finally get targeted to the membrane, not only does it have to go through these nucleotide exchange reactions and GTPase sorting, it also has to be geranyl-geranylated. Now, geranyl pyrophosphate is the precursor to that geranyl-geranylated RAB complex. And what that is, that's the transferase for that. That's an isoprene lipid. Okay, so that isoprene lipid coming from de novo sterol biosynthesis has to be loaded on to that protein for that protein to faithfully finally embed in the membrane. So this is another cycle that has to occur, and only the Rab GDP isoform can be isoprenylated or that is geranyl-geranylated to be specific. Okay, so that's really important. It's another whole component here where lipids are playing a key role. This comes from a 2007 paper from Gisson and Maher. Okay, keep going. A naturally occurring mutation where the arginine is converted to a tyrosine, that's arginine 125, located in the first PTB domain, okay, of that protein we're interested in, the TBC1D1, right, can be linked to familial obesity. So if you have a mutation in that arginine residue, we told you that was important for catalysis. So that means that if that protein isn't working correctly, you can get a rare form of familial obesity. And likewise, that same mutation causes an impaired glucose transport in mice. So you can see that the two are related, right? Now, there's another protein called TBC1D4, which we'll just mention briefly, and it could be also regulated via phosphorylation of those PTB domains, those phosphoryl tyrosine domains. That's what that means. And alterations in protein-protein interactions may occur. Okay, so this isn't the only isoform of these TBC proteins that's functioning this way. Okay, I just wanted to keep you abreast of that. Now, go back to the TBC1D1. Phosphorylation can regulate subcellular targeting of the TBC1D1 rather than actually altering the gap activity of the enzyme. So once again, let's take a look at this. This particular cartoon comes from a paper published in 2011 in Bioscience Reports. And take a look again. There's your vesicle. Vesicle can be transported, for example, to the plasma membrane. There it has an active Rab GTP with an effector molecule. Okay, and that's the activated state. Now, when GAP works, right, like with a TBC protein, don't worry about this other one, what it does, it produces the inactive state. However, with the exchange factor and in association with these other activating enzymes, the inactive form can become active and vesicle transport occurs. This is just another way of looking at it. Likewise, there's a paper that was published online in EMBO a couple years back, 3 years ago. Here we're talking about a P10 protein called PINK1, not related to the specific topic. I'm just showing you the canonical aspect of how phosphorylation controls Rab GTPases, and that controls trafficking of proteins to the membrane. That's why I'm showing you this, all right? So it's just showing you that this is associated with chemiosmotic potential across this membrane, okay? That's important to know. And this happens to be the mitochondrial membrane. At any rate, kinase and or phosphatase, by blocking the phosphatase and supporting the kinase, will alter the Rab8A. Remember, it's the same protein as the substrate for our TBC1D1. And when that happens, when you got this phosphorylated GTPase, it blocks the exchange factor protein, okay? So when it blocks the exchange factor protein, it doesn't allow for GTP to replace GDP. Therefore, you can't make the active form, you see? So this builds up when it's phosphorylated serine 111. Isn't that interesting? That means that this gap activity, okay, is basically still required, even that's what's called activating protein, because you need to recycle to Rab8A GDP to activate for the exchange factor to Rab8A GTP. See, so it's a pseudo-paradoxical situation, so common in biochemistry, where it seems, well, how can that be? It's the opposite of what I might think. It's not the opposite. Remember, these are all cycles. So you have to have a way to replenish all the components of the cycle pathway. And so how do you replenish Rab8A inactive? You have to carry out gap activity, right, which is the GTPase. That's how it works. If you're still confused about it, don't worry. I will hopefully elaborate as we go on. So here we go. Rab GTPase biology. Membrane trafficking between organelles is essential. This is a given, full stop. Ensuring the trafficking works correctly are these Rab GTPase. That's what their job is. Okay. They control membrane identity, like what membrane to go to for a given protein. They also control vesicle budding, like, for example, off the Golgi. and recruitment of these kinases and phosphatases. So they are very important. That last slide just showed you they help recruit, right? There's crosstalk between multiple RAB GTPases and that ensures a spatio-temporal regulation of signaling and trafficking. A spatio and temporal. So you're moving through space within the cell, and it's also a time recorded event. That means there's a denominator called little t. That means the phenomena occur according to time, right? Time is ticking off, and this all is in constant motion. So it's an event that we must record as an event and not as an isolated substance. And that's very important to understand for diabetology. So there's a functional corruption of RAB that can occur, and when that does, it's associated with immunodeficiency, not good, cancer, and all kinds of neuropathologies. So obviously these proteins are important for multiple physiological signatures. So back to TBC1D1. It's a potential molecular regulator of glucose and lipid metabolism. Now, I want you to be disabused with the idea that diabetes is a disorder of glucose. That is indeed the case, that glucose levels are monitored by the pancreas, and insulin and glucagon are controlled by the levels of circulating serum glucose. But I want you to understand that the human is an oleaginous organism, which means we store not carbohydrate against a huge concentration gradient for long-term storage of usable carbon for bioenergetics, and all the intermediate synthesized, including precursors for DNA and RNA and protein and lipids themselves. But that lipid in the form of triacylglycerol storage in the depot fat is the ultimate regulator of all of carbon, nitrogen, sulfur, phosphorus, calcium, magnesium, etc., homeostasis at the atomic level, and then all the molecules and macromolecules you can imagine that are fed by those things. Things like carbon and hydrogen and oxygen, for example, and nitrogen and sulfur. All right, so really important. So this protein's in a linchpin place, okay? So it controls glucose and lipid metabolism, since mutations in that protein, and they do exist, are associated with obesity, I already told you that, and insulin resistance in humans. So obesity obviously has to do with putting on depot fat. So you can see right away, you're not just talking about carbohydrate. TBC1D1 is a member of this RAB-GAP family, that is GTPase activating proteins. And it's activated, as we've said, by AMP kinase and AKT signaling, which are regulated by insulin. And AMP kinase, of course, is regulated by muscle contraction, and also by this nucleotide intermediate called ACAR. Okay, so there's an intermediate in purine biosynthesis called AICAR. Okay, and that intermediate in a biosynthetic pathway to make a nucleotide also regulates this phosphorylation effect and ultimately this GLUT4 translocation. It's been used actually as a molecular probe, ACAR has, to be able to induce this without using insulin. Okay? So you don't have to have an intact insulin receptor and lots of other things which could perturb the purposes of the experiment. Now here's another important, very cool thing. Inhibition of the macrophage, PPAR gamma. Now that is peroxisome proliferal activator receptor protein gamma. I know that's a mouthful, but I've talked about it multiple times in VeraMed, so I'm not going to excuse myself for any malfeasance. Anyways, PPAR gamma, right, which is a transcriptional factor, is one that regulates lipid metabolism, particularly beta oxidation of fatty acids. So inhibition of a macrophage, that's an immune cell, dependent gene expression of that PPAR gamma, contribute to ACAR elicited reduced inflammatory responses to modify lipoproteins and saturated fatty acids, which occur when you have oxidized lipoproteins, oxysterol lipoprotein-laden structures that are in the serum, such as oxy-LDL, and saturated fatty acids, which also can induce that inflammatory response if they're at high levels because they poke holes through membranes. So PPAR gamma is a nuclear factor fatty acid receptor, as I just said, been implicated in energy homeostasis and in many pathological processes as well when there's a mutation or a lack of activity or overactivity. PPAR gamma modulates lipid homeostasis by enhancing the expression of the fatty acid beta oxidation gene family. So it turns on transcriptionally those proteins which will become enzymes in beta oxidation of fatty acids, utilization of fatty acids to produce energy, and ketone bodies. So there's metabolically active sites in the liver, macrophage, adipocytes, and muscle. It's all over the place. It also acts as a regulator of inflammation, as it's PPAR gamma. So there's the link to the TBC1D1 protein. TBC1D1, in fact, inhibits the expression of beta oxidation genes, so it may have an effect through PPAR gamma. All right, finally, let's introduce the paper. Skeletal muscle is a metabolic tissue regulating glucose and lipid homeostasis. Think of it as a reservoir. Glucose uptake requires an intact insulin signaling cascade that induces the translocation of GLUT4 to the cell surface of the muscle. That's the plasma membrane. Hopefully you're fully aware about by now. This regulation of skeletal muscle lipid metabolism, which probably you are not familiar with, has been linked with insulin resistance. So when you have dyslipidemia, you get insulin resistance. And I've talked about this many other talks, some of the mechanisms that we've detailed sufficiently. If you go back and listen to this, I'm not going to be able to cover that in this talk. Too much to do. Potentially, it's caused by excess intramuscular accumulation of bioactive lipids, though, such as diacylglycerol, which turns on kinases, and of course the sphingolipid ceramide. Okay, and they can interfere directly with insulin signaling. Again, go back and watch some of my previous talks in a lot more detail, or you can type those into a PubMed search and you can get a lot of papers from it. Intramuscular lipid accumulation, therefore, attributed to an excess of cell surface fatty acid transporters, okay, leading to an enhanced rate of fatty acid transport. Because of the high levels of fatty acid transport into the muscle, for example, you start overloading the enzymes of beta oxidation. And when you do that, you can shut down fatty acid utilization. These free fatty acids can then build up intracellularly and extracellularly, and that can cause inflammation. That's one of the mechanisms of how obesity is linked to muscle degeneration and to overall glucose dyshomeostasis, because the insulin receptor is corrupted because of fatty acid utilization, which then corrupts the insulin-dependent protein uptake because GLUT4 can't be translocated because of these bioactive lipids, among other things, can cause inflammation. So intramuscular lipid accumulation is attributed to an excess of cell surface fatty acid transporters, as I just said. That leads to enhanced rates of fatty acid transport, but not a concomitant increase in beta oxidation. Evidence that improvements in skeletal muscle beta oxidation can improve insulin sensitivity are well established in the literature. So let's take a look at this now from a more simplified point of view, at least more simplified in terms of not looking at all the proteins involved. There's GLUT4 as a cluster. That's when it's most active. So it goes from the monomeric state to the cluster state in the plasma membrane. So what happens if you want to look at it from insulin-dependent point of view? Insulin binds to the receptor. The insulin-responsive substrate then helps phosphorylate P13 kinase, which phosphorylates phosphorylates 3,4,5 trisphosphate, which then activates a PDK, another kinase, which then phosphorylates AKT, which then phosphorylates AS160. If you remember, one of the earlier slides showed that, playing a role in this vesicular trafficking. Phosphorylated AS160 then controls the movement from RAB-GDP to RAB-GTP. Okay, so that's really important. All this is regulated, ultimately making this sequestered vesicle right here, right? This GLUT4 vesicle, which then allows it, once this occurs, this induction occurs, RAB-GTP activates this movement of the vesicle that we saw in the previous slides to fuse with the membrane. High enough levels of GLUT4 monomers start to cluster, and when they do, you get fully active glucose uptake. Okay, all right. Now, ultimately, it's got to be removed from the plasma membrane when insulin levels start to drop. That happens via endosomal processes, which can be sorted, and sometimes you get complete breakdown of this, or you just get resorting back to these vesicles, which then can be activated once again with another pulse of insulin. Okay. Here's this granulation. I want to just point out that in the early stages of steroidogenesis or cholesterologenesis, this is a precursor to cholesterol, geranyl pyrophosphate in particular. And when you inhibit that with statin drugs, you inhibit this pathway because you cannot make geranyl pyrophosphate from preformed cholesterol because it's a precursor to it. Degradation process of cholesterol is not generated, and you don't get it in the diet, and it's not essential anyway. But statins can drop the levels of that important lipid, isoprene lipid. When that happens, you cannot geranylate this system. If it isn't done, you can't use the exchange factor and make the protein active. And the whole cycle gets corrupted. This is a paper from Nature Reviews, Molecular Cell Biology, as you can see there. Okay, so that's really important. I'm the one that put in this part about inhibited by statins. This paper didn't discuss it, but I do. TBC1D1 is a negative regulator of beta oxidation. Okay, negative regulator. Keep that in mind. Reductions in the protein itself, total net amount of protein, as well as that mutation from arginine to tyrosine in the TBC1D1 increases the rates of fatty acid oxidation in cells and in skeletal muscle, respectively. So, if you reduce the level of the protein or you mutate it so it no longer has the GAP activity, what you get is an increase in fatty acid oxidation, which if you already have a dyslipidemic environment, that would be a good thing, right? Of course, because you'd be utilizing fatty acid. Now, TBC1D1 content differs amongst tissues. Highest protein expression occurs in skeletal muscle. That's what we're talking about. It's more abundant in glycolytic muscle than in oxidative muscle. That means muscle that's carrying out complete fatty acid oxidation, complete Krebs cycle, electron transport, oxphos, just doing glycolysis. There's more TBC1D1 in glycolytic type muscles than there are in the fully oxidative muscles. Okay? And of course, that makes them most susceptible to the TBC1D1 mediated changes in fatty acid oxidation because it's limiting there, you see. So the basis for altering fatty acid oxidation in skeletal muscle is explained by the coordinated alteration of genes involved in mitochondrial biogenesis, oxphos, oxidative phosphorylation, synthesis of ATP because of the proton transport through the F0F1 ATPase in the inner mitochondrial membrane, processes that can be induced by transcriptional coactivator of PGC1A1 alpha are also involved in mitochondrial induction, and so they are ultimately very important players here at the transcriptional level. So there's lots of things going on here. I'm telling you all that so you know this is a very central player. This isn't some kind of peripheral, unimportant pathway we're talking about. Protein-mediated fatty acid transport in muscle is highly regulated, as you might guess, because I told you can cause autoinflammation if you don't regulate it. And it can contribute to the regulation of fatty acid oxidation itself, of course, because it's a substrate. Among the various muscle fatty acid transporters, we talked about again at length in this Vera Med Studio right here. And that receptor or transporter for fatty acids is called FAT, or more commonly CD36. Used to be called an orphan acceptor, and appears to be a key as it's central to the regulation of fatty acid oxidation. So the fatty acid uptake linked to oxidation makes sense, right? Of course it does. Now here is fatty acid oxidation. I had to get one of these pathway slides in there showing you the structures too, because I love structures, because I'm a biochemist. And of course, this is a lipid pathway, so I love this slide all the more. This is just part of the fatty acid oxidation pathway. Just to remind everybody, I'm sure you're completely aware of it. If I gave you a test, you would know the names of all these enzymes and even the mechanisms. At any rate, I won't be doing that today, but you have a long-chain fatty acid that has been removed from, say, a triacylglycerol. It gets esterified to coenzyme A that activates it via an enzyme called thiokinase, which uses ACP. Now you have a fatty acyl-CoA that's a thioester-linked fatty acid, so it's in metabolic play now. Then the enzymes for beta oxidation kick in. The first is acyl-CoA dehydrogenase, so it's a dehydrogenation. Okay, you got a double bond now for them. You got rid of one of those hydrogens. And in the process, you've made FADH2, flavonoidine dinucleotide, which can be used in the electron transport chain to make 2 ATP. All right. The next enzyme, the trans delta 2 enyl CoA, okay, is going to hydrate this whole system. Okay, so you're going to, after you've done this further oxidation, you've oxidized the fatty acid, you're reducing the FAD. Here's your double bond between the alpha and the beta carbon in the preformed fatty acid. See it? There it is right there. Notice it's a trans fatty acid, by the way. More on that later. This enzyme called enoyl-CoA hydratase adds water to that system. See, there's the OH group and there's the extra proton, okay, the beta carbon. Okay, you got rid of the double bond now. Now you're able to carry out the second dehydrogenation and make and reduce more NAD to NADH and further oxidize. That was the whole reason you did this double bond production, so that you could hydrate and then you could dehydrogenate. That's the very common pathway for oxidative reactions of saturated hydrocarbons. Now that's where TBC1D1 inhibits this enzyme. This enzyme, also known as HAD1, hydroxyacyl-CoA dehydrogenase, so the HAD, hydroxyacyl dehydrogenase, D, that enzyme, in a previous paper that I talked about on VeraMed, actually is inhibited directly by TBC1D1. Okay, so there's another place. So it shuts down fatty acid oxidation when it's functional, so that you don't continue. If you do continue, you make NADH, which can go on to the electron transport chain to make 3 ATP. And you also now have the completely oxidized fatty acid, the beta-ketoacyl-CoA. Another mole of coenzyme A, reduced form, comes in, and you get thiolytic cleavage. Okay, it's also known as an acyltransferase activity, depending on what organism you want to talk about. And that's where you get the 2-carbon elimination at the acetyl-CoA level, that level of oxidation. And the acetyl-CoA can go directly into the Krebs cycle. It can be further oxidized to carbon dioxide, of course, making more NADH and FADH2. But also, of course, the acetyl-CoA can be used not in the Krebs cycle, but in the ketogenesis cycle, making acetoacetate and beta-hydroxybutyrate. And both of those compounds can be sent from, not the muscle tissue, but from the liver, for example, to act as a form of carbon that can make it to the brain and the rest of the tissues, including the skeletal muscle and the cardiac muscle. Chain is reduced by 2 carbons. Now, what I want you to notice here, remember before we were talking about if you have too much transport and not enough oxidation, the oxidation becomes limiting. It's because now this C14 fatty acid, 2 carbons shorter than C16, is going to go all the way to the top of the pathway again and compete with C16 fatty acids and C12 fatty acids and C10 fatty acids and C18 and C20 and C22 of various levels of oxidation or hydroxylation or hydroxymethylation, or indeed just plain old double bonds. So the more fatty acids you pump into this system, the more it gets overloaded. NADH builds up. It shuts down these dehydrogenases because they can't be used fast enough in the oxidative phosphorylation chain, the ETC oxidative phosphorylation chain. So it shuts down fatty acid oxidation. You see the point. Nothing is in a vacuum. You can't say something is completely on or off, and everything is related to everything else. You take those 3 comments, take them all the way to the bank. In fact, take them to the front of the church, and you will be guaranteed to begin to understand biochemistry. Nothing works in a vacuum. Nothing is unrelated to everything else. And whenever you have a redox system, redox systems can be easily overloaded. And when they do, it goes all the way back up a chain of command, regulating all the way at the hormonal level how this whole thing was induced. Previous work has shown that skeletal muscle TBC1D1 could act as a suppressor of lipid oxidation, okay, by altering mechanisms regulating fatty acid oxidation in the mitochondria, or even the transporters. And I've talked about those in previous VeraMed lectures. The 1D1, TBC1D1, reduced basal and 5-amino imidazole 4-carboxamide ribonucleotide. Remember, that's the nucleotide that is used as a trigger to study this. Stimulates rates of fatty acid oxidation and slightly stimulated triacylglycerol esterification. So it's altering the lipid moiety to get it ready for beta oxidation, to be in a poised state relative to stored triacylglycerol in oil droplets, for example, in skeletal muscle tissue. Overexpression of that protein, TBC1D1, decreases the beta HAD, that was the hydroxy acyl-CoA dehydrogenase enzyme I just showed you, without altering anything else. Okay, so that's important. That means that that protein has a direct effect on enzyme activity, as well as, we've been trying to argue, transcriptional activity, maybe via the PPAR gamma or PGC1 alpha. Right, so there's multiple layers of complexity of regulation here. So TBC1D1 affects the activity of beta HAD. That's for sure a paper I talked about actually a couple months ago, actually in the summer, so it's been more than a couple months ago. A reduction in the beta-HAD activity results in reduced rates of fatty acid oxidation, of course, because it's blocking one of the key enzymatic enzymes in the pathway. TBC1D1 then alters the protein content of beta-HAD. Other mechanisms are possible, but highly speculative in the absence of data. So they're saying that maybe this protein does have an effect on the total amount of beta-HAD, but it also might have an effect on the activity. Okay, and so this story is still out. Now, the data that was shown previously in one of my very bad lectures is this. They identified TBC1D1 targeted in mitochondria as beta-HAD, that particular enzyme and that enzyme alone. It's reduced comparably, only 18%, but understand that 18%, that's almost 20%, is enough to control the flux through the pathway. Okay, these pathways are far from equilibrium. So you push them a little bit, they go very fast. You pull back a little bit, they go very slow. That's what happens when you're very far away from equilibrium. In other words, a little nudge is like falling off a cliff. There's a difference between sigmoidal kinetics and hyperbolic kinetics if you follow that sort of thing that I've talked about in enzymology, which I know some of you certainly know from your years of experience. All right. So again, knocks it down, the fat oxidation by 18%. It's enough to totally inhibit the beta oxidation that would be necessary to support fatty acid uptake, which can then induce an immune response, which can trigger insulin resistance. See, it's all linked. Okay, now here's some data from the paper. Results employing a TBC1D1 mutant that lacks enzymatic activity, indeed the mutant does not cause much GTP hydrolysis. There it is. There's the arginine 2. This is a lysine mutant here at 854. So they made a lysine rather than arginine. So it's obviously part of the catalytic domain. Now here, full-length TBC1D1 was able to recognize all the Rab substances it's supposed to. So if you put TBC1D1 in, okay, when you add the protein, yeah, you get GTP hydrolysis at Rab8A, that's the substrate, Rab10, Rab14. Cool. Previous studies already shown in vitro and in vivo phosphorylation of at least 6 serine and alternatively threonine residues on that protein. So that means there's something regulating that protein at the level of phosphosite. In particular, the serine 231 and threonine 590, and in human that's serine 237 and 596, were predicted as potential phosphorylation sites for AKT and AMP kinase in response to stimulation with insulin and muscle contraction, respectively. Demonstrated that phosphorylated affinity purified protein, TBC1D1, is produced in vitro with purified kinases, AKT or AMP kinase. Okay, so you can make the phosphorylated protein by using the kinases. So there's a linearity to what we're talking about. So the phosphorylation of that protein by AKT or AMP kinase does not, however, alter the activating protein component of the TBC1D1. All right? Does something else, and we'll see in a moment what it is. Insulin, ACAR, and contraction-mediated phosphorylation of the serine threonine residues in 1D1 regulate the activation state of the downstream GTPases that's controlling to control GLUT4 translocation. That's a Rab8, Rab10, and Rab14. Remember, these are all involved in that vesicular fusion activity that we saw way back at the beginning. Question that I answered was, what about the direct impact of AKT2, that kinase, that isoform, and the AMP kinase, mediated phosphorylation on the gap activity of TBC1D1 using Rab10 only as a substrate? Guess what? No effect, okay? So that means that the gap activity with a really hot substrate for this whole system, Rab10, it's a very specific substrate for this TBC1D1, that activity on activating it is not effective. So again, that's strange. It's paradoxical, but as we'll see, pseudo-paradoxical. More data. These are details of the phosphosites on the TBC1D1. There's the AKT added. No, yes, no. There's no, where the protein is added, no kinase, no ATP. You don't phosphorylate that threonine residue, you see. If you add AKT only, you phosphorylate that threonine residue. If you add the AMP kinase, you phosphorylate that threonine residue. Okay, so that's the important thing. You just see that the amount of protein doesn't change. Okay, so it's phosphorylation dependent. These are Western plots. So AKT and AMP kinase substantially increase phosphorylation of threonine 590, whereas serine 231 phosphorylated by AMP kinase, but not AK2. So here, AMP kinase and no AMP kinase, you get hardly no phosphorylation of serine. You add AMP kinase, you get a lot of it. So this is an AMP kinase contraction-dependent phosphorylation of that residue. Remember, serine 231 is unique to the threonine 590 in that domain. There's no reduction of GTPase hydrolysis when RAB10 when exposed to the phosphorylated form. We already said that. So whether or not it's phosphorylated or not, you see, by AKT or by AMP kinase, you still get GTP hydrolysis. So it doesn't matter that you're phosphorylating TBC1D1. The activity is unaffected. Now here's where it gets interesting. Phosphorylation of TBC1D1 by AKT2 or AMP kinase increases 1433 binding, but does again not alter gap activity. So if you want to get 1433 binding, you need to phosphorylate the protein, okay? You need to phosphorylate the protein, but it has no effect on GTP hydrolysis, whether or not you have phosphorylation. You see, the hydrolysis is the same as if you have none. That's the whole take-home message for this. So it tells you that 1433, which is a player here, requires phosphorylation, but it's still no effect on gap activity. Wow. And to remind everybody what a 1433 protein is, I know many of you know it's right there on the tip of your 1433 mind, but here we go. 1433 proteins are a group of highly conserved proteins composed of several isoforms that are involved in the regulation of protein phosphorylation, and mitogen-activated MAP kinase pathway. So this protein gets some play in tumor biology. Again, I talked about this protein in several papers in previous lectures. I'm not going to be able to talk about it now. Anyways, this is all from, again, the paper we're discussing, the Mafakari paper, JBC, one that just came out. Data from there. Now, this comes from the Mayo Clinic. More about 1433. You find it in cerebral spinal fluid, and it's indicative of substantial, relatively rapid neuronal destruction. Not a good thing to find. Increased CSF concentrations of 1433 proteins are described in patients with various forms of Creutzfeldt-Jakob disease, bad, some other rapidly progressive dementias, and large range of other vascular, inflammatory, neoplastic, and metabolic central nervous system disorders. Not a good thing to get high levels of this protein. So don't mess with phosphorylation via the GAP proteins or the GTPases for induction of fusion of endomembranous systems. In fact, 1433 proteins play an isoform-specific class switch recombination. Holy smokes, right? They're believed to interact with protein activation-induced cytidine deaminase, for example, in mediating a class switch recombination. So they're actually involved in controlling, for example, the recombination associated with immunoglobulin and T cell receptor biosynthesis. So that's one of the other back doors to controlling the immune response. Very, very important protein. We should have a better name. Somebody should rename it. Activation-induced cytidine deaminase, the one we're talking about here that's regulated by 1433, is also known as AICDA, or AID, is a 24 kilodalton enzyme. It's a little guy that creates mutations in DNA because it deaminates cytosine, and that thus converts to uracil, which is then ultimately the next round of replication is recognized as thymine. So it's a built-in mutator. Okay, so that enzyme, that deaminase, is a built-in mutator. It's believed actually to be an antiviral protein. Look at the virus literature, which I try to avoid because it's sort of banal and boring. But because I'm not a virologist. But that protein may have something to do with control of retroviral integration into the cell because it induces mutations. And retroviruses are more prone to mutations because of RNA. Basically, 1433 proteins help transform a CG to a UG mismatch. The DNA replication machinery recognized the U as a T, and hence you get a CG, which was okay, healthy, normal in the sequence. Bonding cytosine to guanine. Now you have a TA base pair. Bad news, right? That kind of change can then induce a mutation. If you have enough of these, it can cause a complete scrambling of genes. It's one of the reasons it's believed to be an antiviral. Okay. The mutation does not alter phosphorylation of 1433, does not alter phosphorylation, does not alter binding or gap activity. So that's all shown here when you put everything together. Mutation of the PTB domain, okay, it's the phosphotyrosine domain, where it's correlated with familial obesity. Remember that. That's what we're talking about here. That's the mutation. An engineered baculovirus expressing the mutant of that protein was made in insect cells. Purified protein was incubated with gamma phosphorylated GTP, loaded GST RAB10, and the amount released phosphate from that system was determined. That's the GTP hydrolysis. There's no difference in gap activity, again, from that mutation, right? And even when you add back a purified form of that phosphotyrosine domain, no difference. So it doesn't seem to affect, again, the GTP activity. This is showing you that there is an effect on the level of phosphorylation, okay? That, once again, you see here is the wild type. It's not phosphorylated if you don't have the PTB domain. Here is the mutant. Again, if there's no PTB domain, it's not phosphorylated. If you have the protein and you have the AMP and AKT and you have ATP, you phosphorylate. Okay, so that's the threonine residue, and that's the AKT activity, AKT mediated phosphorylation. See no differences. AMP kinase, the same effect here, the same exact thing. But now, TBC1D1 mutant arginine to tyrosine was phosphorylated by either AKT or AMP kinase for 30 minutes and incubated with purified gamma phosphorylated P32 GTP loaded Rab10. The amount released was determined. The non-phosphorylated form and phosphorylated form exhibit the same gap activity. So again, all this discussion of kinase activity and all this regulation of phosphorylation, we still can't find out what it's doing because the gap activity is the same. Remember, the gap activity is kind of like the business end of the TBC1D1. Everything else is regulatory core, right? But something's going on we don't get yet. We'll get it now. Sorry for all this writing. Finally, the function of phosphorylation TBC1 is revealed. It's the IREP interaction. Let's read this. The insulin-regulated aminopeptidase. That's a protein which acts as an aminopeptidase. And I'll tell you what some of its substrates are. It's part of that vesicle that gets put in the plasma membrane along with GLUT4 and transferrin. Yeah. So you're loading an enzyme which can then blow out all kinds of activities once it enters the membrane. So you're adding basically a gunslinger to the stagecoach. You're taking the stagecoach to Dodge, and at Dodge, what's going to happen? Lots of things. Some good, some bad. Maybe it's Wyatt Earp, but not a bad guy. Right? Sorry for that terrible metaphor, but it just came to me. Insulin-regulated aminopeptidase, a membrane protein for vesicles, just said that, colocalizes with that glucose transporter. So how does the IREP correlate with this signaling? To find that out, they did pull-down immunoprecipitation assays with HA-tagged IRAP and flag-tagged TBC1D1. So this is doing here. This is the structure. There's the flag tag. There's a GST flag onto the IRAP, okay, and onto the PTB domain. It's just showing you they have antibodies that they can detect it, okay? Phospho versus non-phosphorylated forms of the flag. All right. This is a GST pull down. Now you're pulling down this GST. You're pulling down the TBC1D1 with the GST probe, right? And there's the elution profile, right? You get nothing except you get no change here between having these kinases for the elution. But if you look at the input, it's there, right? And here you can see that the TBC1D1 serine is indeed phosphorylated by the AKT, okay? And so is this threonine residue, so everything is working fine. Okay, so let's go back to here and discuss it. Okay, so the proteins were phosphorylated by purified AMP kinase and AK2, and magnesium ATP was, of course, necessary. Okay, so you had to have ATP in there. Where's it at? Well, it doesn't show you, but you have to have ATP. Okay, makes sense. All right. As shown in 6b, right, immunoprecipitation of IRAP results in co-precipitation of TBC1D1 in the absence, but not presence of the kinase. Okay, so when you add that, when you add the kinase, you don't get that form. So what does that tell you? That's what it tell you. I already said, I'll say it again. TBC1D1 phosphorylation prevented protein-protein interaction with a known partner of vesicular trafficking. So it prevented getting partnered with IRAP when you phosphorylate. So now finally you see an effective phosphorylation of TBC1D1. It inhibits the association to this IRAP protein, which is basically vis-a-vis the interaction with the GLUT4. Okay. All right. So mapping the TBC1D1 binding domain in IRAP was facilitated by fusing that entire 110 amino acid cytoplasmic tail of the IRAP to the C terminus of GST. We showed you this. What was the structure, the construct look like up there? Put in insect cells, express the TBC1D1. They lyse the cells. They take the proteins, again, phosphorylate them with the enzymes, along with magnesium ATP, because you need that, of course, it's the substrate. The pull down of the GST CI-RAP co-precipitated TBC1D1, okay, in the absence but not presence of AKT or AMP kinase, respectively. Okay, in the absence but not presence of AKT or AMP kinase, respectively. See, you didn't pull it down when you did the elution. So it was there at the beginning input. You don't pull it down. Okay, so that means that there's an interaction with IRAP and this phosphorylated domain of that protein. That's what that tells you, right? That's how the experiment was done. So the pull down of the GST-C-IRAP co-precipitated with TBC-ID1 in the absence but not presence of either of the kinases, respectively. So it looks like the C terminus of IRAP is the interacting domain. That's the part of the protein that's interacting here. Mapping of that domain in TBC1D1 was facilitated by fusing two of those phosphotyrosine domains of the protein, those are amino acids 1 to 131, again, the amino terminus, to the C terminus of GST to yield a GST PTBD. Okay, that's the new structure that they created here. All right, now what happens there? All right, binding of a flag tagged TBC1D1 to a GST IRAP, cytoplasmic form of IRAP, cytoplasmic tail, was abolished by excess PTB, but not excess GST. That's just the protein that's tagging it, and that's in GD. Okay, so that's shown here. Right? GST, no problem. When you have that PTB domain, it knocks out that interaction of IRAP with the TBC1D1, and there's your input again. See? So that's really important. So the binding then is abolished by excess PTB, but not excess of GST alone. So it appears that the phosphotyrosine binding domain of the protein is the co-interacting domain. So now you have both domains. You understand the phosphorylation control of the TBC1D1 at the level of subdomain and with the IRAP at the level of subdomain. Okay, so that's really important. So similar to wild type TBC1D1, the GST-C cytoplasmic form tail of the IRAP co-precipitated with the protein that is mutated, because it's not going to affect this, right? In the absence but not presence of the kinases. So again, when you add the kinases, you get rid of the binding of the IRAP. So that's really important, okay? That's really important. It tells you that that's where the business end of phosphorylation is controlling it. IRAP no longer interacts. It means it's not in that vesicle. All right. Moreover, addition of excess GST-CIRAP did not affect the gap activity toward RAB10. So again, it had no effect on the actual GTPase activity, only on the interaction of the IRAP protein in the vesicle by using these pull-down acids. All right, so we're almost there. So here's the GLUT4 retention model. So you've got a recycling pathway, right, where you have to have an inducer, you have to have an ERC protein, and you have to have specific phosphorylation sites and specific sequences of these proteins so that you make the vesicular trafficking to occur. So let's read this out. In fat muscle and GLUT4 glucose transporter is dynamically retained because it is rapidly internalized and slowly recycled to the plasma membrane. Insulin increases the recycling of the GLUT4. So you get more recycling, right? That results in a net translocation of more of it. The quicker you cycle, the more of its sampling occurs and the more of it gets targeted because it has all the correct cargo in that vesicle. That's basically what this is showing you. And these are the two different amino acid sequences for that protein, fore and aft, that is amino terminal to carboxy terminal, for that protein to function in the membrane. Okay, and there's the epitope that's been used to isolate and study it. So again, GLUT4 retention model, the recycling pathway, fast in both basal and insulin stimulated states, and GLUT4 specialized pathway, which is slow basally but fast after induction of insulin. Okay, contribute to plasma membrane GLUT4 distribution. GLUT4 sorted from the endosomal compartment, the ERC, the endosomal compartment, to the GLUT4 specialized vesicles, the GSVs, right? And an unidentified perinuclear compartment, that's what's over here. We don't know what exactly that is. It's something outside the nucleus, that's all they know. All right. And you get in basal adipocytes, and the TR, by the way, is a transferrin receptor. So transferrin is also recycled to this plasma membrane. You need to recycle transferrin because you need to get a lot of iron in. You need a lot of iron in because you had a lot of mitochondrial activity, which means for those cytochromes, you need iron recycled in and out because different oxidation states of iron can cause its own autoimmune response in muscle tissue, particularly in cardiac. Real problem, which is co-linked here. You'll see in a moment. So the IRAP protein is essential for GLUT4 translocation and residence in muscle and adipose plasma membrane upon insulin-sensitive signal transduction. So it's an insulin-sensitive response. So let's talk about IRAP for a few minutes. Insulin-regulated aminopeptidase, IRAP, is a membrane-bound zinc metalloproteinase. And inhibition of IRAP by angiotensin 4 improves cognition in several rodent models. That was published very recently in American Chemical Society journal called Omega, and there's a citation just a few months ago. Springtime now, it's late fall. So angiotensin 4 stimulates norepinephrine-stimulated melatonin production in pinealocytes, and angiotensin 4 inhibits IRAP activity, suggesting that angiotensin 4 modulates melatonin synthesis in the rat pineal gland. So that has something to do with cognition. All right. This is telling you how complicated IRAP is now. I'm wrapping on IRAP. See? Nothing works in a vacuum. Melatonin protects chronic kidney disease mesenchymal stem cells against senescence via the cytoplasmic form of the prion protein. So prion proteins actually have the endogenous constitutive host prion protein is very important in regulating mitochondrial function. That's what it does. So the prion protein that comes in as a toxic exogenous metabolite, that's a totally different thing. It pirates this process. So it's all happening in the pineal gland. So angiotensin 4 inhibits IRAP activity, suggesting that angiotensin 4 modulates melatonin synthesis in the pineal. Melatonin protects chronic kidney disease, mesenchymal stem cells presence against the senescence via the prion protein constitutive dependent enhancement of mitochondrial function. I found that in the Journal of Pineal Research. Yes, there is one. I'm sure Rene Descartes would like to know that. Melatonin improved neurological deficit score and memory and spatial learning ability after cardiac arrest, perhaps via the association of complex 1 and complex 2, supported mitochondrial respiration. If you remember your ETC cycle, those are the very first stages, NADH oxidase and the quinone pathway, which were greatly increased with the melatonin treatment in association with suppressing protein acetylation and improving mitochondrial function in the hippocampus. I got that from PLOS One just last week, a couple weeks ago now. IRAP and the pineal gland cognition promoting melatonin are inversely related as long as the IRAP receptor is actually there. The retention of toll-like receptors in the ER prevents their activation, and IRAP endosomes are involved in early steps of the toll-like receptor 9 activation in dendritic cells. And IRAP stabilize CpG-containing endosomes, which slow the trafficking of that toll-like receptor, which is, of course, involved in the detection of pathogens towards lysosomes. So putting that receptor into the lysosomes and getting it triggered ultimately the plasma membrane. Thus, endosomal retention of TLR9 via the interaction of IRAP, remember that's a proteinase, with the actin cytoskeleton is indeed a mechanism that prevents hyperactivation of the toll-like receptor 9 in dendritic cells. So here, IRAP actually suppresses, because it keeps TLR9 in the cell, not the plasma membrane, it suppresses a hyperinnate immune response. You see, these proteins are way more important than you might have thought. But now, once you start piecing together how you can get inflammation, insulin resistance, and obesity, they can all be linked to the muscle cell. That's what I'm trying to do for you here. Now, what's the interpretation of this research? Since insulin and contraction-mediated serine 3 phosphorylation of the 150 to 160 kilodalton RAB-GAP TPC1D1 by the canonical kinases, the AKT and the AMP kinase, is linked, with downstream RAB-JTPases, GAP activity sensu stricto may be regulated by phosphorylation. The question then proceeds as to the sequence specificity and molecular functional structural domains involved. That's what this paper was trying to get at. Since GAP domains appear deficient in phosphorylation targets, we already told you that, it's unclear how global RAB-GAP, the whole protein, the TPC1D1, phosphorylation will impact the constitutive GAP activity, so it must be maybe an allosteric conformational phenomenon. They found a tenfold higher apparent Vmax of the full-length TPC1D1 compared with the truncated GAP domain that translated to a 1 order of magnitude increase in the KCAT-KM, which I'll mention in a minute what that is. Could be that the thermodynamic catalytic efficiencies of the truncated domains in the protein underestimate the values of the in vivo reaction domains outside of that GAP complex, and that could contribute to conformational realignment of the GAP and thus enhance enzyme substrate complex, thus increasing the rate of the reaction. Kind of a classical allosteric effect. Now, real quickly, enzyme kinetics. Enzyme substrate has a K1 forward, K-1 backwards, enzyme substrate bound together, and the K2 makes enzyme product, and the K-2 reverses it. And that's what those stand for. So K1, K-1, 2 and minus 2 are rate constants for the reaction. Here's a Michaelis-Menten equation for non-complex enzyme kinetics. There's the two equations that are important in Michaelis-Menten. The initial velocity of the reaction is equivalent to the Vmax times the substrate divided by the Km plus the substrate concentration, where Vmax is maximum velocity and V0 is initial reaction velocity. You can rearrange and say the Km is equivalent to the forward reaction and the backward reaction divided by just the reverse reaction and the forward reaction from the ES divided by the forward reaction from ES to ES. Okay, so that is E plus enzyme plus product back to enzyme substrate complex is minimal. If K minus 2 is really, really, really small. See how that works? So you're saying that that backward reaction is very small. That's why you have to add it here. It's actually a way of rearranging the steady state assumption. All right. Now what's KCAT? Remember they said that this affects KCAT. More general term is defined. It's called KCAT. Okay. To describe enzymes in which there are multiple catalytic steps, which there are here, and possible multiple rate limiting steps, which there are here. The Michaelis-Menten equation can be substituted with KCAT. So Vmax equals KCAT times enzyme total. So rearrange the initial velocity, and you substitute, rearrange and substitute, is equivalent to the KCAT now times the enzyme total concentration times substrate concentration divided by the simple Km plus the substrate concentration. So why do we use KCAT? It's in the units of reciprocal seconds, so it's also called the turnover number because it has a temporal aspect, right? Because saturating substrate concentrations, it actually represents the number of substrate molecules converted to product in a given unit of time. Because basically, it's honing in on the K2, the K plus 2, the forward reaction of the ES complex. Right? Substrate molecules converted to product in a given unit of time on a single enzyme molecule, as I just said. In practice, the KCAT values and not maximum velocity of the reaction are most often used for comparing the catalytic efficiency of related enzymes. We just told you that whole phosphorylation business and the whole effect on the protein affects that KCAT kinetic parabola that is associated with the activation of the GTPases, which ultimately links the GLEAT4 translocation to the membrane via insulin induction. This just shows you the KCAT to Km ratio specifically discussed in the paper, or just actually thrown out there. They expect you to know this. I suppose most of you don't, of course. Why is the KCAT ratio a measure of catalytic efficiency? The answer lies in the free energy of the enzyme transition state complex. It's lowering that transition state. Okay, the free energy of the state has to be low enough, to enable the enzyme to bind the substrate at physiological concentrations, but high enough not to unduly restrict then the second part of the reaction, which is the K2, KCAT. So, but basically you change the free energy state by lowering it and make the enzyme substrate complex. Then once again, you go back up with the free energy state and drop it to the enzyme product. The delta G double dagger of the total uncatalyzed reaction is not changed. It's just the delta G double dagger of the catalytic event, which you have truncated, because this is the micro heterogeneity of how that enzyme is coordinated via those two kinetic components. So I want to show you that because I hate it when somebody throws it out and you don't know what it means. Now you do. And you go slowly through that again. In fact, I'm going to give a really detailed enzyme kinetics talk, just freebie, around Christmas time, because I know a lot of people love enzyme kinetics, especially you medical doctors. All right. Synthesis of the system. GTPase activating proteins, GAPs, are potent signal transducers. Even though protein chromatography and polyacrylamide gel electrophoresis suggest that native oligomerization of GAP proteins, GAP domains exist and occurs, the oligomer formation doesn't seem to mediate GAP activity. Okay? These are all like, oh gosh, what if, so what? No, it doesn't seem to matter. The GTPase substrates for the TBC1D1 gap include the following: 8A, 10, 14, and actually Rep28. So there is substrate specificity in the TBC1D1 sequence and domain architecture. We already know that, full stop. Okay, that's another way that enzymes work, is by specificity. Truncated GST tag gap domains, just the gap domains, are similarly active to those substrates, but the full length is more active. That was that KCAT thing we just talked about. Okay, digest that. This means that some substrate specificity related to the kinetics of the activation residues in protein domains outside of the gap domain. That's what it tells you. Oh, yes. Specificity of phosphorylation sites directed by kinase specificity and kinase induction by insulin. Right here. Phosphorylation of serine threonine residues in response to insulin stimulation and contraction has been applied to regulate the TBC1D1 associated gap activity. Okay, we're just wrapping up here. 2 sites, serine 489 and 590, are direct targets of AKT, and 5 sites, 231, serine 231, 404 of threonine, 590 of threonine, 660 of serine, and serine 700, are all direct targets for the AMP kinase. That's a contraction one. By analyzing immunoprecipitates from the insulin and ACAR stimulated tibialis anterior muscle lysate, that's how these experiments I just showed you were done. A previous study described a total of 7 phosphorylation sites that there were. This lab confirms that threonine 590 was indeed an AKT target, and those other ones are all targets for the AMP kinase. But they think that serine 253, serine 253, and threonine 499 by AKT and AMP kinase couldn't be corroborated in vivo in response to insulin. Okay, so they had some problems with that subtlety of substrate specificity. Now let's use the diametrological analysis of TBCD1 in the current context of gap activity. All right, once again, we know that that protein inhibits that enzyme, that dehydrogenase, in the fatty acid oxidation pathway. Remember I told you you're talking GLUT4 translocation with this protein, but you're also talking about the regulation of fatty acid metabolism in the muscle, sensu stricto, related to the TBC1D1, the phosphorylation by AKT, and AMP kinase, which means also controlled by insulin and by muscle contraction. That's it, right? All right. Now, recollect, the TBC1D1 affects the activity of beta-HAD, that's the dehydrogenase. A reduction of beta activity results in reduced rate of fatty acid oxidation. Okay, you would guess that. TBC1, that was the 18% thing we talked about, because it's sigmoidal and the fact that it has a tremendous effect on cell activity. TBC1D1 alters the protein content of beta-HAD. Other mechanisms are possible, but would be highly speculative in this process. So we don't really know. It could be still be a transcriptional phenomenon, but it also may have something to do with the actual dynamics of the enzymatic activity. We don't know. Or maybe the translocation of the protein to the membrane. I would guess that, actually. A 1D1 target of mitochondria is the beta hat. It's reduced comparably, as we said. Future studies need to consider the role of that on the actual activity in different muscle tissues. So causes of metabolic diseases like type 2 diabetes and deficiency that are related to fatty acid oxidation are largely experimentally inaccessible usually. Let me go through this. Beta oxidation is refractory to acquire a complete and quantitative view on the relation between metabolite concentration and flux. Insulin resistance, of course, is strongly associated with free levels of fatty acids. It increases, right, when you get insulin resistance. An imbalance between cellular fatty acid uptake and oxidation, which we were just talking about, leads to accumulation of fatty acids in the cytoplasm, which in turn causes insulin resistance because of an inflammatory response and alterations in the membrane fluidity, which I did not get into. Also observed that a functioning acyl-CoA uptake in the mitochondria is needed to develop insulin resistance. Leading to the hypothesis that intermediates of fatty acid oxidation are part of the problem. In fact, fatty acylcarnitines, I talked about this before in a very bad lecture, may be the key players there. Since the acyl-CoA intermediates are difficult to measure, conclusions are often based on acylcarnitine levels, as I just said, in the blood, which are interpreted as reflection of the CoA concentration of the mitochondria. Yeah, that's a bit of a reach, but we know that they're intimately linked. Similar limitations hamper the understanding of systemic effects of enzyme deficiencies in fatty acid beta oxidation and their impact on global energy and glucose regulation. So this is just like, by the way. Basic biochemistry of fatty acid oxidation reveals complex interactions because it's cyclic. In each cycle, the acyl-CoAs compete for each other, even the shorter versions. The shortened acyl-CoA product competes with the substrate for a set of enzymes with overlapping chain-link specificity. I said that about 20 minutes ago. Complete breakdown to 8 molecules of acetyl-CoA requires 7 reaction cycles, right? There are only 4 dehydrogenases, so they become rate-limiting. As I say there, it results in competition of the 2 pathways, depending on chain length of fatty acids. Competition generates a feed-forward inhibition in the network. The more palmitoyl-CoA that enters the pathway, the more enzyme molecules they occupy, not available for shorter chain acyl-CoAs downstream, so you get a blockage. Not even talk about the NADH building up. The 2 parallel pathways of the breakdown of enol-CoA molecules are of different nature, as we didn't mention, but I have in previous talks. Here it is. Enol-CoAs are converted by a sequence of 3 individual enzymes known as crotonase, medium short-chain hydroxyacyl-CoA dehydrogenase, which is the M-SHAD system, and the medium-chain ketoacyl-CoA thiolase, that's the one that finally breaks off the acyl-CoA, or via the trifunctional protein, which is basically is redundant for these enzymes, but they're all bound on one polyprotein. It's unclear how all those enzymatic systems and the interaction between the trifunctional and the individual and discrete ones interact or compete. It's still not clear. So there's the AMP kinase and AKT, and additional kinase specificity for the phosphoaddition to the TPC1D1 gap activation dynamic directly affect this oxidation pathway. This is a diabentological question and necessarily requires an understanding of multiple pathway interactions in various tissues and organs as time reveals ontologies of these biochemical events. So the paper suggests that phosphorylation, subsequent binding, does not affect gap activity, since TPCD1 targets the GTPases associated with glutathione vesicles. The question arises if it's regulated by phosphorylation. Indeed, the 1D1 does interact with glutathione vesicle-independent protein IRAP in the phosphorylation-dependent manner. So remember that IRAP is linked to insulin-dependent glucose uptake. So the gap activity of the phosphorylated 1D1 fills in the signal transducing pathway and may lead to tumor-associated lipogenesis even, okay? So here's a simplified version here, right? Insulin-mediated intracellular signaling, getting the GLUT4 IRAP up there into the membrane. This puts everything together here. I'm not going to go into detail, but it involves a control over glycolysis, which is called the Warburg effect, the utilization of glucose phosphate to make reducing power in the form of an ADPH, utilizing pyruvate to secrete citrate once it combines with OAA, getting a neoplastic fatty acid synthesis. So this is called the lipogenic switch, and you can see these are where these journals come from. This is telling you there's a direct link to this tumorigenesis. I'm not going to go into detail with this. I will in a subsequent talk. Likewise, the insulin-mediated aminopeptidase occurs in single-spanning transmembrane zinc metalloproteinase. And the cysteine aminopeptidase of placental leucine aminopeptidase are known as PLAP, and those are basically the same enzyme. IRAP was initially described in the adipocytes, being localized with GLUT4, as I said. Insulin stimulates the vesicles to relocate and fuse. Since IRAP is distributed more ubiquitously than GLUT4, it's apparent it possesses other functions, and here's where it's showing you where it is. IRAP includes the N-terminal amino acid residue of several small bioactive peptides, in fact. It includes the metin, Lu, and enkephalins, dysnorphin A, neurokinin A, somatostatin, cholecystokinin A, oxytocin, and vasopressins are all really important peptide hormones. Whew! So, in fact, disruption of the hypothalamic pituitary thyroid axis in rats with breast cancer through an oxytocin IRAP system can then induce high levels of serine oxytocin and a reduction in this serum intermediate. Okay, so that's really important. Likewise, if you look at the PKP, AKT pathways, phosphorylating the AS160 so that you get it bound to 1433, that's all linked to IRAP. That's going to associate with RIP11, which is also associated with the RAB GTPase, which is further signal transduction for the GLUT4 sampling system. The IRAP does this, okay? It affects these various hormones, right? The IRAP affects oxytocin, angiotensin, and there was the third one here. There's just aminoproteinase. Okay, so all three: vasopressin, vasopressin, angiotensin, and oxytocin. Each of those are substrates for the IRAP. Okay, so this is also going on whenever you translocate the IRAP to the plasma membrane. I want you to understand that's what its function is. So let's go back to this paper. TPC1D1 interacts with GLUT4 vesicle resident IRAP in a phosphorylation-dependent manner. Co-precipitation occurs with both full-length IRAP and the 110 amino acid cytoplasmic tail, and the interaction is reduced after phosphorylation, such that the interaction is blocked with an excess of the PTB domains. However, the binding of IRAP does not affect GAP activity, so that kinase cascade is responsible for signaling the activation of resident GTPases. So the zeitgeist is that phosphorylation prevents recruitment of TPC1D1 to the RABs located in GLUT4 vesicles. That is, the RAB GTPases cannot be activated because of their subcellular control over co-compartmentalization, which is mediated by the phosphorylation of the GAP agency, that is the ability to carry out the activating protein in the TPC1D1 polypeptide. So it never gets there when you alter that phosphorylation cascade. So here's the synthetic a posteriori. The mutant coding TPC1D1 was associated with increased risk for obesity. The mutant could alter ligand binding of the first PTB domain and the AMP kinase interaction, but no evidence of that was shown. There was no difference in phosphorylation pattern, GAP activity, or phosphorylation-dependent IRAP. We saw all that in the data. So where does it leave us? Basically, that the 1D1, which is a GAP, becomes phosphorylated by AMP kinase and AK2, and when that happens, the interaction with another player, this IRAP, shows up. It's lessened, the interaction is lessened, and this has the effect of diminishing the assembly of the GLUT4 vesicle for subsequent transport to the plasma membrane upon insulin signaling. So this hyperphosphorylation, TPC1D1, decreases glucose uptake since insulin is over-signaling, and thus the cell becomes resistant. And that's exactly what insulin resistance looks like. Now, what does the evidence say from publications analyzing the system? Reported that regular exercise can diminish obesity-induced insulin resistance. And does it by enhancing GLUT4 translocation. The literature suggests physiological insulin signaling and promote GLUT4 translocation of the plasma membrane. Okay. Exercise-induced muscle glucose transport involves AMP kinase and the calcium calmodulin kinase and the so-called atypical protein kinase C. Also, AKT is a key factor. Phosphorylation of AKT serine 473 is temperature sensitive and increases in temperature dependent manner. Wow. Exercise-induced increases in body temperature uniquely induces AKT signaling, but not AMP kinase signaling, which is controlled by contraction, in obese-associated type 2 diabetic rodent skeletal muscle. So perhaps temperature elevation during exercise improves insulin signaling, right, by activating the AKT, which could counteract global insulin-resistant associated hyperphosphorylation of downstream substrates like the TBC1D1 due to a competing hierarchical inhabitation of those multiple phosphorylation loci, which restrict the phosphorylation caused by AMP kinase activity or AKT activity by overstimulation with insulin. Put into perspective, then, the evidence points to a diabetological mechanism controlling glucose uptake via insulin signaling that depends not only on the kinase induction and phosphorylation of key gap protein isoforms and aminopeptidases, but further how those events are spatially and temporally executed. Postscript, one more paper from Huang, restoration of myocardial heart muscle cell glucose uptake facilitated by the glucose transporter 4 translocation contributes to elevation of diabetic cardiomyopathy in rats after a duodenal jejunal bypass. Okay, don't worry, you'll get this. This is more diabetology. I'm telling you, it's complicated because that's how cells actually work. This is from the Journal of Diabetes Investigation, came out a month ago, 2 months ago now. Almost. The duodenal jejunal bypass, or the DJB, common surgery for obese people, reduces diabetic cardiomyopathy, or DCM. How's it do it? The DJB cardiac muscle reacts differently in T2D, that is diabetic patients, associated cardiomyopathy relative to GLUT4 translocation. This is known. The surgery restores systolic and diastolic cardiac function, remits cardiac hypertrophy, cardiac fibrosis, and it actually lowers atherosclerotic lipid deposition and which is unrelated to body mass. Wow, how to do all that? The mechanism of the surgery suggests a surgery-dependent upregulated myocardial insulin signaling via hyperpolarization by AKT substrate of the AS160, all right, and TBC1D1 by facilitating GLUT4 translocation and restoration of glucose uptake, but that was associated with less CD36 fatty acid transport. This is why the bypass gives you insulin sensitivity, because you start decreasing the amount of fatty acid uptake. So here, the diabetological mechanism is in the diabetic myocardium serves to limit excess fatty acid uptake while co-enhancing glucose uptake to drive myocardial contraction. Rather than a spatiotemporal event ontology paradigm, here there is a differential fuel supply event ontology from fatty acid to glucose, you see? Still linked to the gap-mediated signaling. Now, I put all there at the end. I know that's a lot, but I did that so that you start to see full on what I'm trying to persuade you is a major paradigm shift in biochemistry, and that is diaventology. We have to look at these things in real time, over time, sometimes extended periods thereof, different tissues, and then the homeostatic control based on hormonal regulation, subcellular regulation of kinases, fuel utilization, and ultimately the final physiological processes that can be regulated or dictated by minor fluctuations in the relative amounts of the proteins that are synthesized and regulated post and co-translationally by lipid modifications and signaling. Thanks a lot for your attention. This is Dr. Daniel Guerra from Vera of Med saying adios. This last slide here is St. Jerome in his study as a painting by Antonello de Messina. St. Jerome, I like this painting. It's there. He's working. He's like a scholar in the Renaissance period. He's got a lot of the parts of this painting here, which was done on wood, shows you a lot of scholarly things. which sometime later on I'll point out to you why these paintings are full of this. But I'm not St. Jerome. I'm just Dan Guerra. There's my Facebook page. There's my email address. There's the company. We look forward to hearing from you. And anything you want to say about or talk to me about this presentation, I look forward to that as well. All right. So you have a good beginning of December, and we will be talking next. I don't know if I'm going to do the enzyme kinetics talk or maybe something else. There's a lot of other things on my palette that I've put together, and you can be sure that this December, just like December in the past, I'm going to be giving you guys a lot of lectures, free lectures, so that you get a lot more Vera med at your home during the holidays. So all I can say for right now is bye for now.